Good afternoon, everyone. We're team IZJU-China. So today we're thrilled to present our project, FoCas. So we only have short 15 minutes, so just focus on our project. So we have never expected before that our story would begin in a small and common office in an infectious department of a hospital. We were talking with an infectious disease doctor when he received a call from the nurse. It was bad news. A patient with SA pneumonia wasn't getting better, even though they used higher doses of antibiotics. The doctor signed. His kidneys and liver are already under heavy stress. We've tried combinations, but that's all we can do. That sight was imprinted in our hearts. Shocked at first for such a common and serious issue, antibiotic resistance. The main treatments are now still toxic, expensive, and limited. So then, as the number of published articles describing novel and fancy treatments keeps mounting, the infectious patients are still waiting and suffering in pain. Countless patients need a low burden and effective therapy to overcome drug resistance, which continue encouraging us through all of journey. So this kind of bad news can be heard all around the world. And as we research further, AMR mainly arises from the existence and transmission of resistant gene, which will encode antibiotics interfering proteins. We also interviewed the pharmacy director of a top hospital in Hangzhou, who told us the new MRSA drugs R&D is low because of the suitable protein target hard to find, and their delivery systems widely varies. Inspired by all above, we finally decided to design our little fox into two systems. So the first one is CRISPR-Cas9 system to eliminate the drug-resistant gene, and DNA origami to deliver our drugs in the cells. So DNA origami used long single strand of DNA called scaffold and a set of short strands called stables. Our system is designed to tackle drug resistance and the transmission of the gene. So here's the flow. It has many advantages as a drug delivery system, including self-assemble, programmable, and full addressable to the functional components. DNA is also easy to be degraded without causing any cytotoxicity or immune reactions.
In MRSA, as an example, the mecA gene encodes the PP2A protein which is low affinity to antibiotics. Then after evaluating the significance of mecA gene and the effectiveness of recovering the MRSA to MSSA as a potential therapeutic strategy, then we can decided to use the MRSA as a model pathogen, and we can use the mecA gene as our model resistance targeted gene. Firstly, for our project, we used cat nano to design the platform with M13, MP18 scaffold, and 192 staples. Then we used OxView by checking the 3D shape, relaxing the structure, and eliminating the geometric strain to visually understand the stability and integrity of our DNA origami platform. Next, we monitored the RMSD, distance and energy analysis, to simulate the physical behavior and confirm multiple equilibrium. The wet lab also verified the successful formation of a rectangular nanostructures under the atomic microscope. Then, as this is a very stable platform, then we designed to add some more functional components in our platform. This includes the membrane permeabilization component and the loading component. Firstly, for the loading of the sgRNA and Cas9 complexes on our DNA origami, we organized 6 sites of the PAM-cap staples. And this kind of PAM-cap staples, they can capture and connect with the PAM-rich sequence. This PAM-rich sequence, they can recruit the Cas9 protein. On the other hand, a linker sequence was added to the sgRNA sequence to connect the sgRNA to our DNA origami platform. By these 2 sides, our sgRNA Cas9 complexes, they can load it to our platform, and once inside the cell, the naturally existed RNase H enzyme, they can cleave the linker sequence and release the sgRNA Cas9 complexes. For our wet lab experiments, the loading of the sgRNA and Cas9 complexes were verified by UV spectroscopy. And secondly, for the membrane permeabilization component, we added the G4 sequence to the staple strand, and to recruit the heme, the incorporation of the G4 heme complexes, they can act as a kind of enzyme that catalyzes the environmental hydrogen peroxide to generate reactive oxygen species. The Rose molecules, they can oxidize the proteins and lipids of the cell membrane and destroy the cell membrane. With the formation of the small pores of the cell membrane, our drug can directly enter into the cell. So for this component, firstly, we must confirm that the generated Rose molecules, they will not affect the drug itself. So by developing a diffusion model, we estimated and tracks the movement of the Rose molecules that's surrounding the DNA origami, and the result indicated a relatively low damage probability. And secondly, we also started to de novo build a transmembrane delivery model, and this model, it will be optimized following our presentation, and the final version will be shown in the later. For our wet lab experiments, firstly, we have verified the successful loading of G4 heme by the UV spectroscopy without any obvious dimerization. For the effects, firstly, we use the APTS assay to prove the catalytic activity of the G4 heme complexes. And secondly, we use the NPN assay to verify their transmembrane effect with a higher fluorescence corresponding to the experimental groups. Then to start our project, our HP groups has interviewed and discussed with our nurses to discuss about our potential application scenery, and we investigated and identified the wound infection as our target potential application scenery for MRSA. As this, you know that the wound environment is a very complex environment that's filling of multiple RNA or protein enzymes. So these enzymes, they might damage the sgRNA and Cas9 complexes. So we should have a component to protect our cargo. To address this problem, we organized 12 sides of the locking strand on both sides of our DNA origami. This strand, they are complementary to the opposite strand, and also we added a SIL modification in the middle of this strand to form a dice-4 bond. And of these structures, they can lock together and to roll up into tubular structures to protect the cargo. For the assessment of our protection function, our dry lab modeling also used some probability models to evaluate the probability of these enzymes that can transmit through the pore and damage the inner cargo. There are 5 enzymes that are taken into consideration, including 4 kinds of DNA enzymes, 4 kinds of protein, and 1 kind of DNA enzymes.
Up to now, the basic model has been built, but we are still striving for the optimization. During discussions with clinicians, they expressed major concerns about safety, especially about our targeting where resistant bacteria without harming normal cells. To address this problem, we integrated a targeting model onto FoCas, Aptamer. DNA aptamers are short, single-stranded, and oligomerization titles that fold into distinct three-dimensional conformations that specifically bind to target molecules. We introduced a short complementary sequence to both aptamer and stem post to link aptamer onto origami. Our dry lab developed a workflow for aptamer design. We input the target protein, PBP2A, into aptate trains to generate an initial aptamer candidate library, then perform the first screen. After that, ZDoc and HDoc are applied for cross-validation. Finally, they are combined with existing aptamer database like AptameDB to get the final result. LSM imaging and mean fluorescence intensity measuring in wet lab confirm a significant target effect. Our wet lab has also found through NPN assay that membrane permeability evolved after the loading of aptamer, indicating aptamer may function as an anchor to facilitate membrane permeability. This is the second time that our transmembrane model has been optimized. By this time, all designs for our FoCas systems have come out with full functional models. As I mentioned before, the sgRNA Cas9 and the DNA aptamer, the G4 hemion, and the locking strands. Following experiments were conducted to verify the effects of the whole system. Here is the final version of our transmembrane model. It validates the multi-process of developing, including drug anchoring, ROS forming, and transmembrane diffusion. We inferred and measured how large the holes will be formed and whether the diameter of the holes allowed the drugs to enter step by step. These simulators also provide visual panels to be user-friendly, like the left and the right and the middle. And to confirm internalization in wet lab, the complete structure with aptamer shows significant intracellular success by LSM imaging and MFI analysis. To validate the efficacy, the appearance of white clonies demonstrated successful intracellular cleavage of vaccine gene in E. coli.
So now we have completed our wet and dry lab designs and validation. However, we didn't just stop here. We also explored our application scenario by drafting a looser structure to deliver drugs through BBB. We also plan to modify the surface with PCD to improve the stability and also validate the vessel entry. And through questionnaires and interviews we carried out, we find that the public awareness of gene editing is still limited. So we carried out some education to the public that synthetic biology can be safe, controllable, and beneficial in fighting AMR. Through the whole process of our human practice, we did not forget the sign of the doctor. So if we bring the jug, our little fox, to market one day earlier, then patients can be saved one day earlier. We are pushing forward our practical application to a FoCas. We also cooperated with some other iGEM teams and to compile the safety handbook. And also we carried out an ethical matrix. So our little fox has taken its first step, and we do hope that at the end of the path, we will embrace a resistance-free future and wish we will never hear the heavy sound for the doctor again. And at last, I want to share with you something, because actually we booked our early flight back to China because we never thought that we'll be in the finalist. Yeah, so making here was a totally shock for us. And we had to change our flight last night at about I think 11 p.m. Yes, but we're still glad it means that we got to miss our flight and meet all of you today here. Thank you.
Congratulations again, team IZJU-China.